Hi there. We've recently uploaded a video that demonstrates how you can count neurons in microscopic bright field images fast. That's a very useful thing to do because the number of neurons in an image, of course, is a very simple but valuable readout that isn't immediately available from bright field images. Today we're going to talk about a somewhat more complex analysis, the segmentation of neurons in images, by which we mean the detection of cells and parts of cells in an image like this. Marked in blue are the cell bodies or somata, marked in yellow are the neurites. Image segmentation can of course be done either using classical computer vision algorithms or using the machine learning framework of your choice. Of course, we use our Vader system's built-in functionalities. Vader has a neat UI for labeling images, which is machine learning lingo for teaching the machine how to segment images. For this particular task, it took only 3 images to get the performance you just saw. Now, once you've segmented your image, what can you do with that? First thing you can do is compute relative areas. Like, for example, the number of pixels in this image that are either blue or yellow to the number of pixels that are black. This will give you effectively the confluency of this image. Or you might compute the ratio of the area covered by neurites to the area covered by cell bodies. That's the ratio of the number of pixels that are yellow to the number of pixels that are blue. The nice thing about that is that neurite area is proportional to neurite length. And we've actually gone and validated that by tracing all the neurites in this image by hand. We determined the factor between neurite area and total network length in an image to be able to estimate one from the other. To get an idea of the relative error we're making when estimating network length from neurite area, we did a small resampling simulation where we determined both numbers for smaller sub-images and then combined those numbers, added them up for virtual full images. We found that the relative difference between the measured network length and the scaled number of pixels is just plus or minus 4% at most. So that tells you that whenever you need the network length, you can just use the numbers of pixels that are segmented as neurite in an image. Or you can combine the neuron segmentation analysis I'm talking about here with the neuron counting analysis I was mentioning earlier. You can, for example, compute the average neuron size by dividing the whole neuron covered area by the number of neurons. Finally, you can use segmentation to compute cluster sizes. This is the distribution of the blue blobs in this image. This peak in the distribution here is for single neurons. Trust us, we've checked that. And that gives us a possibility to estimate for each blob how many neurons are there in this blob. And that gives us this distribution here. All of these are just a few examples of the possible readouts you can get from image segmentation. It's a very powerful method that can give you all kinds of biologically important information on your cell culture. Confluency, of course, is a general marker of cell health. Neurite growth relative to the number or size of neurons can be an indicator of differentiation, for example, when you're deriving your neurons from iPSCs, or of course it can be a toxicity marker. And a lot of cells like to cluster. Depending on your cell type and your application, it may be a very good sign or a very bad sign when your cells start clumping together. Analyses like these can be easy to implement, for example, on our Vader system, and they're a great way to get quantitative, objective, uniform readouts from bright field images of your cells. Using bright field images means you don't have to stain your cells. And not having to stain your cells means you can get those readouts while your cells are still being cultivated. It also means that the time and money that goes into staining can go into more productive things, doing more experiments, doing more comprehensive experiments with more treatments, more cell lines, and more conditions altogether.